Otherwise, I'll go back to this view. Let me know now when I change slides. Can you see the change now? Can you see them changing now?
No, ma'am, not yet. No.
No, you cannot. Okay. Okay, then let me present it like this itself.
Yes, ma'am, it's fine. It's absolutely fine.
Okay, so as I was telling everybody that, human health has 3 intervention strategies, including vaccines, diagnostics, and therapeutics. And diagnostics plays a central role in the containment of infection and management of public health. And we have a large number of diseases that are of concern for our country, including AIDS, tuberculosis, malaria, typhoid, chikungunya, dengue, to name a few, and of course, COVID. And when we talking of tests, we're looking at tests that have a short turnaround time, are less expensive, and can be implemented with limited infrastructure in resource-poor settings. And such tests, which, if possible, would improve compliance, would increase the implementation of the testing programs across the country, even in the peripheral settings, and augment various disease prevention efforts that are being made by the government and different agencies. Therefore, simple, rapid tests are the need. And such rapid tests that have acceptable sensitivity and high specificity of detection would be useful in testing settings where counseling or first aid has to be given along with test reports. They are extremely useful in settings such as emergency rooms, where we need to have results immediately before any interventions can be planned. Also, as I was telling, in resource-limited settings where supplies are scarce, like in the developing and the underdeveloped nations, we need rapid tests which can be performed with little instrumentation. And for initiation of timely therapy for the infected people, like pregnant women, and for initiation of preventive therapy for at-risk populations like the healthcare workers, rapid tests have great benefits. So we're looking at tests that are quick to perform, easy to do, require little or no instrumentation. The people who have to perform these tests can be trained minimally. And if these tests can be performed non-invasively, that is on samples like finger prick blood, urine, they will be highly beneficial. In the last several years, there has been a lot of development taking place for rapid tests. And it is the availability of these rapid tests that fulfill one and many of these criteria that has allowed testing to move from big cities and high-end labs to clinic-based settings to primary healthcare centers and come to the reach of one and all. And one such technology for rapid tests is what is the talk for today, which is NEVA, Naked Eye Visible Agglutination, a technology that allows rapid detection to be done using finger prick blood. And the basis of this technology, which is very simple to perform, is very user-friendly and gives rapid results, is based upon use of recombinant antibodies. So before I move on to describe the technology and how it works, for the benefit of all the students that are there in the audience, I would just like to explain some basic concepts. Antibodies, all of you must have read about them, heard about them, are Y-shaped molecules, which are produced by our immune system. So these are glycoproteins that are made in response to the presence of a foreign molecule or antigen in our system. And the human body is capable of making thousands and thousands of different antibodies that can bind to different antigens and protect us from a plethora of diseases. And it was very early in 1890 that the German physiologist Emil von Behring first demonstrated when he took the cell-free material of blood from a guinea pig that had been infected with diphtheria and injected this cell-free material into another guinea pig who was infected with diphtheria. And he was able to protect and save this guinea pig with this cell-free material. He also showed that this cell-free material was very specific and it could only protect against diphtheria. It could not give the protection against tetanus. So it was Behring's this discovery of the curative powers of blood that led to the first treatment for diphtheria and tetanus way back in 1900. And Behring called this natural antidote as antibody, for which he received the very first Nobel Prize in 1901 in physiology and medicine. And following his work, there was an unprecedented progress and achievement in the field of antibody research. And it led to several Nobel Prizes being awarded, some of which being to Porter and Edelman in 1972 for the antibody structure and function, to Onegawa in 1987 when he told us how the genes for antibodies are arranged in the genome and how they rearrange and recombine to give the enormous diversity which we witness for different antibodies. In 1960, Burnett and Medawar discovered the mechanism of tolerance. And then Kohler, Milstein, and Yern were awarded the Nobel Prize for giving us the technology for making monoclonal antibodies. So these magnificent antibodies are Y-shaped molecules which comprise of two chains, the light chain and the heavy chain. The light chain comprises of two domains, the variable and the constant, while the heavy chain comprises of the variable linked to three constant domains. The arms of these Y-shaped molecules are capable of binding the antigen, while the stem of the Y is what gives the effector functions like binding to the phagocytic cells, binding to the complement, and other molecules. The variable domains of both the light and the heavy further comprise of more conserved regions, which are called the framework regions. And these framework regions provide the scaffold for the hypervariable CDRs or the complementarity determining regions, which are the primary contact points for the antigen binding. So it is the variable part of the antibody which primarily binds the antigen, and the constant parts provide the scaffold and the effector functions. The variable of the light and the constant of the light form the light chain. The variable of the heavy with the first constant of the heavy chain forms what we call as a FD fragment. And it is the light chain and the FD which form the Fab fragment. So this bivalent antibody can be digested with enzymes like pepsin to give what is a Fab2 molecule, or with papain to give the Fab, or they can also be made recombinant. And the smallest antibody fragment that can bind the antigen is what we call as the single chain FV, which comprises of the variable domains linked by a peptide linker. So while the native antibody, which is a complete antibody molecule, is bivalent, that means it has two valencies it can bind to 2 antigen molecules, one by each of the arms of the Y. Similarly, the Fab2. The Fab and the SCFV are monovalent. That is, they have only one antigen binding site because that Fab is only one arm of the Y. So they are monovalent. They have only one antigen binding site that can bind. So a Fab or an SCFV fragment can bind a single antigen molecule, while the native antibody can bind 2 antigen molecules. And it is this Fab fragment which has been used to develop reagents for the NEVA technology. And the first development of this technology was done for HIV infection, and therefore what was developed was NEVA HIV, or Naked Eye Visible Agglutination Assay for HIV. So NEVA HIV is a whole blood based agglutination assay which allows on-site detection of HIV infection. And this NEVA HIV uses recombinant molecules which are Fab fragments to which HIV antigens have been attached. And these Fab fragments are derived from antibodies that can bind to human red blood cells. So the NEVA HIV reagent is basically the Fab fragment of an antibody that can bind to human RBCs, and the Fab fragment of this antibody has been linked genetically to sequences for HIV antigens to form a Fab HIV antigen fusion protein. So this reagent essentially comprises of 2 components: an anti-RBC antibody Fab fragment and the HIV antigen. So we developed both the components of this reagent. We developed 3 antibodies that were universal and could bind to all human RBCs irrespective of the major and the minor blood groups. So these antibodies were not blood group specific. They would bind to all human RBCs, whatever be the blood group of the blood sample. So 3 such antibodies were developed: B6, 10F7, and A41. And based upon the literature, the second component, which is the HIV antigens that could be used for this reagent, were identified. So based upon the literature, we identified 3 HIV antigens that could be used. One was derived from GP41 of the HIV-1 virus. Another was derived from the GP36 of the HIV-2 virus. And the third was derived from the P24 protein of HIV-1 and was named P246. So these are not complete full-length proteins, but there are regions of the proteins against which in the literature it was shown that antibodies are made by the infected individuals. So these are regions of these proteins that are immunodominant. Immunodominant means against these regions, the infected people make a lot of antibodies. So we took these 3 HIV antigen sequences and we made fusion proteins with the antibody Fab sequences. So for this, genetic engineering was used, and the sequences encoding these HIV antigens, which you can see as P246 or envelope 2 or envelope 1, they were genetically fused. That means their coding sequences, their nucleotide sequences were fused to the coding sequences of the FD fragment of different anti-RBC antibodies. So the sequences for the light chain and the sequences for the variable of the heavy and the first constant. As I told you, that's called the FD fragment. So the sequences for the FD and these different antigens were cloned together in different plasmids, and they were expressed in E. coli. So E. coli cells were transformed with the plasmid that would express the light chain, Separately, the plasmid expressing the FD chain or the FD HIV antigen fusion was transformed so that we could get the FD chain. And once we had the light chain and the FD chain expressed, this light chain and the FD were allowed to mix together and renature to form what we call as a Fab molecule. So the light chain and the FD would associate, and the disulfide bonds would allow a Fab to be made in vitro in test tube. So like this, we made Fab carrying the HIV-1 specific antigen, we made Fab carrying the HIV-2 specific antigen, and Fab carrying the P24 antigen. And these were then formulated to make a control reagent. So the control reagent is where we have only the Fab for the 3 antibodies. The HIV tails are not fused. And then we have the test reagent, which is the Fab to which the different HIV antigen tails are fused, like the HIV-1, the HIV-2 specific tails, and the P24s. So we made a control reagent and a test reagent formulation using a mixture of these proteins, and we use them to develop NEVA HIV. So now, how would these proteins allow us to detect the presence of HIV infection? And this is how these reagents work. So here we have the NEVA HIV reagent, which comprises of the anti-RBC Fab with the purple HIV antigen tails. When this reagent is added to blood from an HIV uninfected individual, then these anti-RBC Fabs, they will go and bind to the red blood cells because that is their property. They're supposed to bind to the red blood cells. So these Fab molecules, when they're added to blood from an uninfected individual, they go and coat the red blood cells. And these HIV antigen tails are hanging like this. However, as I told you, Fab is monovalent. It can only bind to a single antigen. So these reagent molecules will go and bind to the RBC surface, but because they are monovalent, they can only bind to one RBC. They cannot bind to 2 different RBCs. So they cannot cross-link RBCs, they cannot give agglutination. But if the blood is not from an uninfected individual, but from an HIV-infected individual, then this blood, besides the RBCs, it will also have anti-HIV antibodies, which that infected individual is making in response to the HIV infection. So these anti-HIV antibodies, which are being made by the infected individual, these are natural antibodies which are bivalent. And they can bind to these purple tails in the NEVA reagent. So when you add this NEVA reagent to the blood from an HIV-infected individual, these Fab molecules will go and bind to the red blood cells. And these light blue antibodies, which are made in the infected individual, they will bind to these purple tails of the HIV antigen. And because these antibodies are natural antibodies which are bivalent, they can bind to the tails of the RBC, coated RBCs of 2 different RBCs. And thereby, they will cross-link the RBCs, creating a mesh leading to the clumping of RBCs and what is seen as agglutination. So there'll be no agglutination if the blood is coming from an HIV-infected, uninfected individual, but there'll be agglutination if the blood is from the HIV-infected person. So this reagent can lead to clumping of RBCs, which is seen as agglutination if the blood is coming from an HIV-infected individual. So this is the concept of the test. So these reagents were developed, and then they were used to develop the assay. So the assay which was developed had 3 spots. One was a control spot, one was an HIV-1 spot, and another was an HIV-2 spot. So on a glass slide, the regular glass slide on which we do the blood smear or the bacterial smear, the same glass slide is used. We add a drop of blood to each of the spots. We add a drop of control reagent to the control spot, a drop of HIV-1 reagent to the HIV-1 spot, and a drop of HIV-2 specific reagent to the HIV-2 spot. And then we mix the blood and the reagent and we observe it for agglutination. And this is what you will see. For example, in this, we see clumping in the HIV-1 spot. That means this blood sample is coming from an individual which is infected with HIV-1. Here, we see clumping only in the HIV-2 spot, indicating that this blood sample is coming from an individual which is infected with HIV variant 2. And if we see clumping in both the spots, it indicates that there is co-infection of HIV-1 and HIV-2 taking place. So this test reagent was developed, and then it was used to check the efficacy of the test in comparison to several other US FDA approved tests that were in use. And we found that NEVA HIV performed as well as the other US FDA approved tests and was able to detect HIV infection as good as them. Then this test was evaluated on clinical samples in the Indian population across the country, and more than 1,200 samples were tested, and it was found that NEVA HIV had very high sensitivity of detection. It detected all positive samples as positive, and it was highly specific in detecting HIV infection. So this technology of NEVA HIV was released by the Honorable Prime Minister of India, and it led to several publications as well as patents. And finally, this technology was transferred to Cadila Pharmaceuticals Limited, Ahmedabad, who developed this test as a 2-spot assay. So this is the box of the NEVA HIV kit. These are the bottles for the control and the HIV reagent. And this is what the slide looks like on which the test is performed. So this technology was licensed to Cadila Pharmaceuticals Limited, and the commercial launch was done. And this is how I'll show you the actual test is done. So this is the slide on which the test is performed, which is part of the kit, which I showed you. A drop of blood is added to both the spots. The control reagent, you can see the control reagent is added to the control spot. The HIV specific reagent is added to the HIV spot. So this bottle contains the antibody fusion proteins that were made as a formulation. Okay. The blood and the reagent is mixed with a stick, and after 2 minutes, the slide is observed. And this is how the clumping is seen if the blood is coming from an HIV infected individual. So the commercial launch of the kit was done by the Vice President of India, and this technology received several awards from different organizations, including ICMR, DBT, and DST, and also from the different academies, the science academies of the country. And within the first 2 years itself, Cadila was able to solve more than 150,000 tests in the country with no adverse reports. What is most heartening for the developers is when people other than the developers use the technology and show how good it works in the real scenario. And this is exactly what happened. So Johns Hopkins, USA used NEVA HIV along with 2 other tests, 2 other rapid tests, OraQuick and Determine. Determine was developed by Abbott. OraQuick was built by another US company. And both these tests are US FDA approved tests. What Johns Hopkins did was they purchased NEVA HIV from the market. They purchased these two tests from the market, and they used these two tests to check if a two-step algorithm of rapid tests can be used to screen pregnant women for HIV. So for HIV, let me tell you, it has been shown that HIV virus gets transmitted from mother to fetus. However, just before delivery, if the pregnant woman can be detected for HIV infection and drugs, HIV treatment can be given, the transmission of the virus from the mother to the fetus at the time of delivery is highly reduced. So in such a setting, what is required is a rapid test that can give quick result and thereby allow the drugs to be given to prevent this transmission of the virus from the mother to the fetus. So Johns Hopkins was looking at developing a two-step rapid test-based algorithm that could be practiced to allow pregnant women to be tested and prevent this transmission to take place from the mother to the fetus. And they published their work in the International Journal for STD and AIDS. And we came to know of this when we read this report. So we were not aware of their study. We were not aware that they were procuring these kits from the market and using them. We only came to know that they had used NEVA HIV when we read this report. And the data which they showed is summarized here. So you can see that each of the three kits was used as a first test and in combination with the other two as the second test. And all of them gave highly specific detection. But as you can see here, whenever NEVA HIV was used along with any of the other two kits, either as a first test or as a second test, the sensitivity of detecting infection was higher than when NEVA HIV was not part of the algorithm. So this told us that NEVA HIV was improving the sensitivity of detection in this setup, and it was performing at par and even better than the US FDA approved kits. Because whenever NEVA HIV was part of the two-step algorithm, it improved the sensitivity of detection. That is, it improved the number of positive cases being detected. So we were very happy that NEVA HIV, which was being sold in the market, was performing well, and it was helping in detection of HIV and curtailing the transmission of HIV. And this technology was also mentioned in his address to the nation on the Science Day by the Honorable President of India, Dr. APJ Abdul Kalam, where he told how scientific progress being made in our country was enriching the society and helping people. So we made more improvements in the technology. We improved its production capacity to reduce the costs, including more variants of HIV which came up gradually, and that led to several more publications and patents. And having seen the benefits of the technology, we then envisaged to use this technology for the detection of malaria. So malaria, all of you would be knowing, is a disease which is caused by the parasite Plasmodium. And Plasmodium infects the red blood cells. And the infected red blood cells, they show the presence of malarial specific proteins on their surface. That is, Plasmodium infected RBCs have Plasmodium specific proteins that come to be present on the RBC membrane. So if we can detect such infected RBCs, it would indicate infection with Plasmodium and thereby malaria. So for this, what is required is a bispecific antibody. So unlike the NEVA HIV reagent, which was a monovalent antibody, Fab fragment linked to HIV antigens, Here, this reagent requires bispecific antibodies. That is, antibodies which are Y-shaped molecules, but where the two arms of the Y will bind to two different antigens. They have two different specificities, thereby calling it a bispecific antibody. And again, such antibodies would be made by recombinant means. So these bispecific antibodies would have two arms. One arm would bind to all RBCs, infected, uninfected, irrespective of the blood group. That means it would again be an anti-RBC Fab. And the other arm of the Y would bind to malaria-specific antigens, that is plasmodium-specific proteins that are present on the surface of infected RBCs. So here we have a bispecific antibody reagent. When this reagent will be added to the normal blood, that is blood coming from an individual which is not infected with plasmodium, again, it is only the blue which can bind to these blood RBCs. So these blue would bind, but because these RBCs are not infected with plasmodium, the green will not find the binding antigen and therefore it will not bind. So again, these bispecific antibodies, when they're added to normal blood, they would not cause agglutination. However, if the blood sample is of a plasmodia infected individual, that means a malaria carrying individual, then these RBCs would also have the plasmodium specific antigens, the green antigens on their membrane. So these RBCs now can be bound by the green arm of the Y because they are specific to the plasmodium antigens. So when this bispecific recombinant antibody reagent is added to blood from a plasmodia infected person, then the two arms of the antibodies would bind to the two different antigens, one being the universal antigen on all RBCs, the other being the plasmodium specific antigen on the infected RBCs. And this would lead to cross-linking of RBCs and clumping, which is seen as agglutination. So by using this bispecific antibody molecule, we would see agglutination if the blood is coming from a plasmodium infected individual, but we would not see agglutination. There would be no agglutination if it's coming from an uninfected individual. So by developing different kinds of recombinant antibody-based reagents, we can detect different infections in different ways. All these reagents are made by genetic engineering, and while the making of these reagents uses state-of-the-art science, the use is very simple, very rapid, and visible by the naked eye, requiring no instrumentation. In 2020, COVID came. And therefore, we thought of extending this technology to develop NEVA COVID. That is a rapid test to detect COVID infection. In COVID, literature told us that antibodies are elicited in the infected individuals against all the viral proteins, but there are two proteins, the spike protein and the nucleocapsid protein, against which the antibody titers are very high. So epitopes or regions of the spike protein and the nucleocapsid protein were identified against which the antibody response is found in most individuals. And as you can see here, that some of these S epitopes and the N protein epitopes show very good reactivity against sera obtained from COVID infected individuals. They are highly specific to COVID because sera from individuals infected with other coronaviruses, which cause seasonal infections, do not show any reactivity to these epitopes. Also, the healthy contacts, that means people who are uninfected do not show reactivity to these epitopes. And these epitopes also don't show antibody reactivity in people who were infected with the first SARS infection that happened in 2003. So that means these epitopes are highly specific to COVID and they do not show cross reactivity with other infections by other coronaviruses. So these antigenic sequences were used to develop NEVA COVID. So these pink tails that were earlier of HIV specificity were changed now to antigen sequences that were specific to SARS-CoV-2, which is the causative agent of COVID, to develop NEVA COVID. The concept is the same, that these anti-RBC COVID antigen fusion proteins, when added to blood from uninfected individuals, would not show agglutination. However, when they're added to blood from COVID-infected individuals, they will show agglutination because of the presence of anti-COVID antibodies in the infected individuals. So these reagents would allow the detection of antibody response to COVID in the infected individuals and thereby indicate infection with COVID. With advancement in technology, from the times when NEVA HIV was developed to now when NEVA COVID is developed, other systems and technologies have also improved. And therefore, the Fab fusion proteins that we earlier made in E. coli, in bacteria, can now be expressed in mammalian cell culture. So here we have the light chain and the FD. We have the light chain and the FD vectors being co-transformed in CHO cells, that is the Chinese hamster ovary cells, in mammalian cells, and the Fab proteins or the Fab antigen proteins are expressed and secreted through the signal sequence into the culture supernatant. They are harvested, recovered, and then purified to get the Fab proteins, as shown here. So this technology, which is NEVA, is simple. It has its virtues. It's a completely indigenous technology which we have developed in the university with the infrastructure in the lab. And it is a technology which uses whole blood. It doesn't require serum or plasma isolation, and thereby it becomes extremely rapid, can be performed in less than 5 minutes. It is very simple to perform. It can be performed with very minimal training. It requires no instrumentation, no electricity, not even a drop of water from outside. And therefore, it can be performed on site in all areas, in peripheral areas of the country, in a primary healthcare center. And that is why it becomes a point of care test. That is, it can be performed at the site of care. And because it uses reagents that are recombinant, it is cost effective. It doesn't require the use of animals to prepare serum or antibodies. And it can be applied to many different diseases. So that is what is NEVA technology. And as you can see, it uses a lot of genetic manipulation, recombinant DNA technology to make those reagents. But once those reagents are there as a formulation, as a kit, it is very simple to perform. And that is what I want all of you to understand, that we have to use science. We have to do high-end work. But we have to make tools, we have to make products that are simple to use, that can be used by one and all, and that can be for the benefit of everyone. And therefore, on the Science Day, I want that all of you should take a pledge that you, as students of science, will do work, will do research that can benefit the society, that can reach the society and help mankind. And let me tell you one thing, that we can only reap the benefits of science when we work hard. As you always would have heard, the deeper the roots, the greater are the fruits. You need to have deep roots if you want to benefit, if you want to enjoy the fruits of science. It's very important for you to develop skills, to develop versatility, to be able to develop the skills that are required to analyze, think new, dream new things, and then do things that will be benefit of the society. And therefore, with this mandate, Professor Dinesh Singh, when he was the Vice Chancellor, established this center. So the center was established to carry out research in the area of infectious diseases. And not only do research, which is done everywhere in the university, in the country, across the globe, but also to train students, train teachers, train scientists to do work, to develop skills that can help curtailment of disease and infection. And that is why the center, SYNAPSE, has the mandate to not only carry out research, but also to train and interact with industry so that academia and industry can work together and develop technologies for our country. Our Prime Minister launched the Make in India campaign. He launched the slogan, Made in India, Made for the World. And we must all strive for it, because it is only technologies which will take our country forward, which will help us to put our stamp across the globe. And for that, all of you need to build the skills. And we at this center try to do that. We try to do that by our outreach activities. We want to promote science. Therefore, we invite colleges. College students come, they see the infrastructure, they interact with the people working in the center so that they can have a feel of what kind of work is done, what kind of things are possible, what are the new technologies that are coming up, okay, and how they can use these technologies for their work. We hold training programs where BSc students come, where MSc students come, and they learn different techniques, different technologies that are being used day in and day out to do different kinds of things, and which would be very useful to them wherever they go to do science, whether in India or outside India. So we hold workshops. Earlier, before COVID, we were doing workshops in the winter breaks and the summer breaks so that students could come and attend those courses. Let me tell you, these are all hands-on courses where each and every student is working on the bench and doing things with his or her own hands. So it's not demonstrations. It's not about just telling you. It's about doing hands-on courses, learning those techniques and learning them to the extent that you can go and do them anywhere. So it's not that you get the results one time. It is something that you will learn and will be with you forever. So we hold these training courses. BSc students come. This is about one such course where people from Shivaji, Shraddhanand, Venkateshwara, and different colleges came and they did the course. We also hold teachers training courses so that teachers can come to know of the new technologies, teachers can come to know of the recent developments, and they can incorporate these things in their teaching so that they can teach the students and get them exposed to more new things. So these teacher training programs are also hands-on. The student training programs are also hands-on, where students are learning on the bench, doing things with them, so that they can get exposed to different technologies and they can get interested in science They can get interested in doing things with their own hands and learning new things. We'll be holding such workshops again. We're starting again now because now the COVID wave has subsided. So in March itself, we are beginning. We're going to be starting with these courses. They're courses meant for BSc students. They're courses meant for MSc students. I will share this with the SYNAPSE team so that if there are students who are interested, they can join, they can do these courses. These courses are not paid. We get funds from the government of India to do these courses. So we do them so that interested students can work. And let me tell you, all students who have come to these courses, they've been here from morning 9 to evening 6, happy, very happy doing the work, getting the results. Not even once have they said that we would like to leave. Can we leave now? And they only say, please do let us know when you do the next course. We want to come again. So on this Science Day, all I want to say is that take this opportunity, take the time, use your time wisely to learn more. There are a lot of skills, there's a lot of information on the internet which you can imbibe. There are handout skills that are coming, that are being done in different places. Please use this time of your BSc to learn more and more, get exposed to different things so that you can use them in your science and do good work. Thank you. I'll be happy to take any questions.
Thank you, ma'am, for this really wonderful session. So there's just one question that is there from our side that you told about how this technology was first introduced for HIV, but has now been expanded for COVID and malaria. So could you please tell us if there's any more research going on for expanding this technology to other infectious diseases?
So I'll tell you, right now we've been very busy with COVID. Okay, as was told by in the introduction also, we're also developing therapeutic antibodies for COVID. Okay, which is like you people might have heard of a antibody cocktail being developed by Regeneron, which also became available in India. Okay, so these are neutralizing antibodies that are able to neutralize the virus and prevent the infection from spreading in the individual. So these are therapeutic antibodies. So there's a lot of work. We're busy with that work right now. Okay, for COVID. Okay, there was also some work for tuberculosis going on. But yes, this technology is widely applicable for a large number of diseases, and therefore it's not that we will give up on it. So in the near future, we'll have more things, more applications of this.
Thank you, ma'am. That was extremely wonderful and encouraging to hear. We can also see Dr. Vijay Chaudhary, sir, with us today in this meet. So, sir, would you please come and speak a few words? It's really an honor to have you here with us today.
So you need to unmute yourself.
So, in fact, I had to go somewhere, so I was driving. So, rather than going to the lab, so I'm sitting in the car. So, in fact, all this training to Amita comes from me. And in fact, I would say not comes from me. My wishes have been sustained. Because, after doing PhD from Delhi University, Patel Chess Institute, I went abroad to NIH and did antibody engineering work. And many of those are first in the world. But all those work was related to developing therapies for the cancer, immunotoxins. So I was the first one in the world to make recombinant immunotoxin. And that has become a very big technology. And a product has come out in the market by that lab, by Mentor's lab in 2018. But what I did was when I came, and because I'm born and brought up, I've studied in Delhi University. So I knew what are the limitations. But without worrying about the limitations, I worried more about what is required for the country. Okay. So cancer, anybody can work on cancer. People are working on cancer. But unless you develop a good diagnostics or you develop a good therapeutics, nothing will work. it will end up only in publications. So another thing which I learned abroad was, if you do good work, I've got enormous number of papers in Nature, PNS, but should I continue doing that kind of work, which is not very easily feasible in the country, or should I use my knowledge, my skill, into something which is useful for the country? So that is what exactly I did. I did not pursue, not even once, what I was doing abroad. Okay, so that what will happen with people like me, who have resources to go abroad, I would have gone every year outside for 3-4 months and would have continued that kind of work. And never ever the NEVA HIV would have been developed, never ever hepatic antibody. So it is 100. We are very proud. I'm very proud. Not when I say I means the entire lab. Amita is one of the senior most contributors there. There have been many others, that we could do whatever we wanted. And we could do whatever we wanted, and not to be followed by anyone. And I also inculcated the training in all my students, that they do such a good science that you get into a good publication. And any good publication, a meaningful publication, meaningful is very important, has a patent worthiness. And patents may not be useful for a personal benefit, but I tell you, it is for personal benefit also. But the national wealth is created only because of the patents. Papers do not create the national wealth. It is the technologies supported by the patents which becomes the national wealth. So that, and because of that, not only we publish very well, we also file the patent appropriately. So I'm really grateful to my students who made many things, because Amita has covered only one aspects of the laboratory work, which has been very successful and has been admired by everyone, including, you see, Kalam Saab admiring the work. It gives so much of pleasure, So I'm so happy that I could attend it and I could hear the entire, and I really admire efforts of the students. I think it is a students organized function, and I congratulate your teacher in charges or even the principal who are behind the scene. Thank you so much. Thank you. I hope you enjoyed the lecture. Yes. Thank you.
Thank you so much, sir. Yes, sir. It was lovely listening to you. Your inspiring
Actually,
words.
actually, yesterday I gave a lecture. It was yesterday or day before yesterday. I gave a lecture for similar students at Ravlan Lanan, but the topic was very different actually. So maybe that topic, sometime I can repeat because that lecture is ready. It is because you must have seen Amita has touched upon one part of the antibody role of antibody, that is diagnostics. Antibodies are very versatile molecules. So I just gave, I think that it was day before yesterday. So because we want to promote training on antibody, and that is what that SYNAPSE is trying to make, and we'll be doing the advanced courses also on antibody. I mean, that may be too advanced for undergraduate students, but there are lots of people, even from the industry or who are doing MSc or PhD, they may be interested. So very well, all the best to all of you.
Thank you so much, sir. And thank you so much, ma'am. You explained it so well. And I really could understand a lot of things. And I'm definitely going to take a lot of things from this lecture, from this webinar. And I hope so our participants here who are present with us today. And ma'am, one of the participant is asking, how would if they're wishing to join the workshop, then how do they contact you, or is there any website or any form that they need to fill for the workshop?
Okay, so I'll just tell all of you, there is you need to go to the website for SYNAPSE. Right now it's under some updation, so it may take a couple of days, but nonetheless, since I have the Synapse ID or Swati's ID with me, I will share the workshop flyer once it is there. Okay, and we are planning this in the mid-semester break, which happens from 12th to 19th of March. Okay, that is the tentative plan, though the dates are yet to be finalized, but it will be somewhere around that. So as soon as the flyer is ready and the schedule is ready for the workshop, I'm going to share it with Swati, and I hope, and I would request her to share it with all the students and the teachers in different departments so that they can circulate it and students can benefit from this. There are limited seats in each workshop because, see, it's purely hands-on. As I said, each and every student will be working independently. Okay, so it's a very intense hands-on workshop. There are limited seats, but these workshops are done very routinely. Okay, so if you don't get an opportunity in one workshop, do not lose heart. The next workshop, the next workshop. So they keep happening, and you'll be able to get. Okay.
Yes, ma'am. Thank you so much, ma'am. I would now like to request all the audience present here to please fill the feedback form, the link of which is shared in the chat box. I would like to request Damayanti, the co-editor of Synapse team, to please present a vote of thanks on behalf of the entire Synapse department. Over to you, Damayanti.
It's my privilege to propose the vote of thanks on this occasion. I, Damayanti Dasgupta, the co-editor at Synapse, on behalf of the entire union, would like to thank our speaker, Dr. Amita Gupta ma'am, for gracing this event and sharing with us your valued opinions. Thank you, ma'am, for your precious time and for your cooperation. We all are inspired by your words. You helped us learn about this very relevant and useful technology in detail. I would also like to thank Dr. Vijay sir for addressing us. I would like to take this opportunity to thank our principal, Dr. Vijay Lakshmi Nanda ma'am, for providing us the opportunity to organize this webinar, Dr. Nisha Vashish ma'am and Dr. Yasha Yadav ma'am for their support and guidance. I would like to extend my thanks to the President, the Vice President, General Secretary, event managers, and all other union members of Synapse for successfully organizing this event. Last but not the least, I would extend my gratitude to the audience for being there throughout this session and making it a successful event. Thank you all for being there today, and thank you once again, ma'am and Dr. Vijay sir, for gracing us with your presence today. Thank you.
Thank you, everyone. Thank you. It
Thank
was a
you,
wonderful
everyone.
interaction.
Bye-bye.
Thank you.
Thank you so much, ma'am. Thank you so much, sir.